Hello, everyone. See if I can get close enough that you can actually hear me. Hi. We are now to our last session of the day. For some reason, we always start with like big picture and then with all the molecular stuff, and we always end with behavior. And so it feels like Warren and I are at the end of the day often. So thank you for those of you who have hung in there, and thank you to all of our presenters. As always, I'm in awe and very humbled to be amongst this group of scientists and this group of families. So I'm going to talk to you about a study that I've been working on for the last, I guess it's been about 8 years since we started this. It was kind of initiated by some parents that said, we want you to study our babies. And that got us going. And then it took a while. Now we have some funding and hopefully we'll be able to get more and keep this going. One of the things that you keep hearing us talk about is how important it is to have information from a lot of families to get the picture across the whole range. Warren's going to talk in a little bit about work that he and I have done over the last 30 years about the syndrome, the shared symptoms of people who have primary agenesis of the corpus callosum. So we're looking at, in our work, people who don't have a lot of other problems in the brain, other pathology, and who don't have hyperplasia. They have agenesis, and typically we're looking at isolated agenesis. So in that group that Elliot was showing you that does the best, the mild deficits or the almost normal, what we've been trying to understand is in those people where functioning is optimal, how close is it to typical and what are the challenges? With the presumption that the system's using these backup mechanisms, it can probably do really well at its best and probably has limitations on a day-to-day basis. So we've been trying to map out that syndrome in adults and teens. But as parents are often asking at these conferences, what about everybody else? What about all these kids? What about the little ones? What about the range of disorders? So we're now trying to really address that. And a lot of this is motivated by the fact that we can capitalize on brain plasticity by intervening early in life. The brain continues to change throughout maturation, but those first 5 years, a lot can change. So in other fields like autism research, there's been a lot of work that says, yes, early intervention matters. You can diagnose this condition, callosal dysgenesis, before birth. So before birth that intervention is probably going to be helpful. The earlier we can do that, and the earlier we know what the challenges are and how to recognize those challenges, the earlier we can start to intervene. So we're doing 2 studies. They're called 2 studies, but let me clarify this. The infant study, which is from 6 months to 36 months, is the funded study. So if somebody can join our study in the time window when we're enrolling people and they're 6 months or 12 months old, they can be in that study. But that leaves a lot of people out, right? Because it's just this time window. So in general, we have the child study, which is really kind of the infant and child study. So all of that data is going to be pulled together. So even if your child is 15, they can enroll now and get tested and contribute to this data set. And it's all comers, anybody that pretty much the range of diagnoses. There are a few things that we do kind of isolate out. This work, particularly the infant study, is conducted with colleagues at University of Minnesota, and I will talk a little bit more about that in a minute. So the general study design is also, we're trying to optimize access to all of you and your access to us. And so this is done remotely. We're relying on parent interviews, both verbal interviews like over the phone and a lot of parent questionnaires. in time, Dr. Richard's lab, our lab, Warren and I are working on computerized tasks that we can do remotely as well. And so we'll be expanding what we're offering and the data we can collect. But you could participate from anywhere in the world. So the infant study is, like I said, enrolled if you can enroll before 13 months of age. It has parent reports 6 months, 12 months, 18 months, 24 months, and 36. So the first 3 years of life. And then we carry on and we continue every year until someone's 18, collecting the same kind of questionnaires. And I will tell you in a minute more about what's involved. We're also, for the infant study, we have the money to collect saliva, which we get the saliva samples from the child and the 2 biological parents, and then we ship them off to somebody who can do something with them because I can't. So they are currently going to San Francisco. But again, the value, we're going to hit you over the head with this if you haven't gotten it yet. The value of collaborating is that if I've collected your saliva samples and I send them to Dr. Scherer, he doesn't have to collect them. Or if he has collected them, I don't have to collect them. So we're trying to also minimize the pressure on all of you by you do it once and we share information. So we're collecting saliva samples. We're also then getting your MRI scans. If you can share them with us, and those Dr. Tovar Moll's lab is reading. So we're getting MRI, genetic information, and behavioral information in all ages and following longitudinally. The other thing I wanted to point out about this study, and I'm just really pleased about this and excited, is I'm not a developmental psychologist, I'm a clinical psychologist. And most of my work was really focused on teens and adults. So the question came up, how do we characterize infants? Not my thing. But we had a postdoc in our lab who worked with one of the premier autism researchers who had this huge autism study looking at infants. And now Jed Elison, is at the University of Minnesota and is running the Baby Connect Home study and has infant studies on autism and various things. So he and I sat down and said, how do we go about this? What are the right tests to use? What's the best procedure? Because you've been doing it for 10 years and we haven't. So tagging on their expertise and on their comparison groups. So in order to be able to know anything, we have to be able to say people with this diagnosis compared to people without the diagnosis, what's typical, what's not typical. Well, that adds a lot of work and a lot of money and a lot of time to collect all those typical kids. They felt them. So one thing I will say is when I was saying baling wire and chicken wire and duct tape, it makes you be really thoughtful and thinking through how do we optimize what we're doing, collaborate with other groups, and get the most bang for our buck out of all of this. So all that said, when we started out this collaboration, they are testing kids who are at risk of autism, infants, because they have a sibling with autism. And then they track them and find out, are you at high risk for autism and you develop it, or are you high risk for autism and you don't? They also had a low risk sample. Since we started this collaboration, they've also added Down syndrome and Fragile X. And I believe they got other groups they're adding in. Fernanda and I have collaborated and worked on what data to collect behaviorally in Zika and in children exposed to COVID. So they will be collecting in Brazil the same questionnaires in those groups, so we can compare across all these groups. So the questions of why are some of the genetics in autism the same as the genetics in ACC, and well, we'll also have behavior to put together with that. So the other 2 groups are these control groups that Dr. Elison has that are just really big studies. So we have all of his expertise, and a lot of the data, which is very exciting. Key questions that we're asking. Come on. There we go. So what do we want to know? I'm going to talk from the infant study perspective first. So questions that our study is focused on are, what are the developmental delays and challenges that are common? What can you expect when you have a child with this? What's the range? Because it's pretty variable, but what's the range of variable? And does the outcome depend on the type of callosal agenesis? Can we associate that, like Dr. Scheer was showing? What about these other conditions? You know, if they have other brain conditions, like I said, in this group, we're including hypoplasia and more brain abnormalities. So we get a bigger picture of what is going on. In what way are the infants and toddlers with dysgenesis like autism, Down syndrome, Fragile X, other kids at risk for autism symptoms and other developmental problems? One of the really cool things that is, because we can diagnose this at birth, we don't have to wait until a child starts to show symptoms of autism to test them. We have this whole group that we know some of them are going to show those symptoms. Some of them are probably going to have attention symptoms. Some of them are going to have other things, but we will have tracked them before those even show up. So we can go back and try to figure out what would have given us a clue. What are the predictors that might help us recognize in other kids that they're more vulnerable to some kind of outcome? And then ultimately, all of this is, the more we understand the what and the how and the why, we can start to then work on interventions. So very briefly, this is our research battery. I'm going to comment on this a little bit more later on. But it includes a lot of things that are focused more on autism kind of spectrum, social behaviors, basic language function, basic developmental milestones. We get a lot of natural history, like how were you diagnosed? When were you diagnosed? What interventions have you done? All of that kind of thing. Repetitive behaviors. And then for this infant study where we have the funding, a portion of the group is actually going to be assessed in person in Minnesota. So we have the money then to pay for people to travel and have clinical evaluation and get some feedback, which is really exciting. And if we get the money, then, like I said, to do some scans there, then we will also be able to add that. So far, and this is the child study and the infant study, but the kids that are in this younger age range, we have over 300 children enrolled, which is really exciting. You can see here that the majority of them purportedly have complete agenesis. Some have partial, some we're not sure. And then there's this dysgenesis category, which we're including hypoplasia and all of the other weird things that come up in neurological reports. The radiological reports where we can't see this, we haven't gotten the scans and it's not real clear yet. So hopefully we will get scans, your MRI people will send them in, and then we can read them ourselves and give a more accurate diagnosis. Also, you see that it's primarily the United States, but around the world, and that we are including people that have no other known diagnosis, and then others that do have some known genetic or anatomic abnormalities, and we'll be able to break those out. I'm going to skip through that. Just a few preliminary results because it's exciting. What we're finding so far, and the orange bars are our comparison group. They're the typical controls. We haven't done any comparison right now with autism or high risk. This is just a huge group of typical kids, babies. What we're finding is that at 18 months, social interaction in our agenesis group is not as good as expected as in the typical group. And that continues on, well, that's 12 and 18 months, that same social competence here in this area. This just indicates, these little stars indicate that it's notably different from the typical kids. If you wonder why these go the different directions, it's because of the way the tests are set up. Sometimes a high score is bad and sometimes a high score is good. And so you'll just have to trust me that these, the way they're set up means that there's some deficit in the social functioning in our ACC kids. What's interesting is by 24 months, they don't seem to be showing the same challenges. Now, we'll see how that holds up as we get larger samples and we get more people in the study. And how does that hold up? Is that just only some of the kids that really get better or seem to meet their peers and some don't? So those are the kind of questions we're asking. This is about diagnosis that would be consistent with autism, pervasive developmental problems. And again, at 18 months, our agenesis group, blue, is showing more of those symptoms, but getting closer, still more at 24 months. We've not gotten far enough along to look at 36, so we'll see. And then language. This is kind of a little bit in a different direction. So at 12 months, there's a little bit of a difference, but they seem pretty close as far as words produced, because most kids can't produce many words at 12 months, right? But as the typical children take off in language production, our ACC kids are lagging behind. How will that hold up over time? We don't know. What we're seeing in gestures is our ACC kids are behind across the board. But that's one of the questions. We're also doing the Vineland, which Jasmine administers over the phone and gets motor information. So are you behind me on gestures because you can't move? Are there motor problems, or is it because of a cognitive issue? So we're really trying to get a broad understanding of how the children with DCC are developing so that we can start to break out what's causing what. So fewer words and gestures across that age range, and more pervasive developmental problems at 18 months and lower levels of social functioning at 12 and 18. That's all our preliminary data. That's a paper that's going to be submitted just any day now. And then we'll start moving forward with the analysis comparing to autism and looking at temperament. One of the exciting things is questions like, does temperament at 6 months predict anxiety at 12, 14, or 12, 18, 24, et cetera? Does temperament and anxiety predict autism and social impairments? So again, that kind of longitudinal questions that we can only do this kind of study. The child study, which extends all the way up to 18, are very similar. Questions are about what are the developmental delays and challenges that you see in this group. How do those challenges change across development? So one of the things that Warren and I have observed and reported on many times is that as children with ACC, or as typical children hit prepubescence and start to think more abstractly, and they start to have more advanced social skills, which also corresponds with the time when their corpus callosum is going through a period of myelinating and is more effective, that our ACC kids tend to fall behind in their rate of development. Now, we've observed it in smaller groups. Is that going to hold up when we start looking at larger groups and we can look longitudinally? Or are there different time points? Again, what can we expect? How can we intervene earlier and avoid some of this? What psychiatric diagnoses? So autism, ADHD, OCD. How many of you have had like smorgasbord of how do we describe the behavior of this child? We'll try to clarify some of that and see what the range is at more detail. And then of course, academics. So all that to say, I'm really excited about the range of what we're able to do. I'm going to jump through this. This is our team. So you'll meet Jasmine in the hall, along with Ella and Brianna, who are here for the summer, helping gather people into to participate. And I encourage you, like I said, you're the choir, go out and encourage people to participate. All of the data that we collect will be shared with all of these other researchers and will become part of this core that people can ask questions of over and over and over again. One other thing I wanted to point out before we wrap up here, in future directions. Again, I'm hammering it home, the value of the IRC5. And so today you met Fernanda Tovar-Moll in Brazil. You met, see, I think Tanya's lab is mentioned in here. She's not listed because she's not the neuropsychologist, but she's got a team of neuropsychologists in Paris, Linda Richards' lab in Queensland, Linda's Richards' lab in Washington University. We've got people in the UK and in Australia and in Italy that are all collaborating and sharing their data. And so part of the planning ahead and what we've been doing when we say we started this organization in 2015 was we sat down, a lot of this was 2017, all of the neuropsych, the cognitive psych people sat down, and across the top here, if you could see it, which is, the point is not really to see the detail, it's just to get the gist, that here are all our groups, here are all these tests that we do, who's doing what, who's done what where, how can we align this so that we all start collecting the same things, what's been done in the different languages. So coordinating all of that so that we use our data most effectively is part of what we've really been doing as far as laying the foundation. So we came up with a battery that says, these are the tests, the questionnaires, and then these are the in-person tests that we all agreed on. These are our goal. We may not always get them all done. We're going to try to do these tests and these people at these levels of developmental functioning, and then we can compare them and share them with everybody else. So that's the agenda research-wise. And I have started listing out the projects and the things that we can do with it. And our group this week was saying, oh my gosh, we are so blessed. We have an abundance of information. And now it's how do we keep hiring people to do the computer work and the analysis and write it up and keep bringing it back to you. But we're just going to keep gathering it. So please, if you haven't already enrolled, please do. And I will stop now and hand it off to Warren so he can talk about adults.
It worked. Woo, finally, at the end here we get. So I'm going to continue some of the thoughts that Lynn was talking about with respect to particularly social and language behavior and competence in persons with ACC. But I'm going to kind of push this towards thinking about elaborative thought and creativity in persons with agenesis of the corpus callosum. Just to re-emphasize what Elliot and Lynn have both said, the group we're working with is persons with isolated ACC, that is corpus callosum absence only or primarily, who are typically higher functioning. Dr. Schur called these persons with mild to no developmental delay, and not dealing with persons with what we might call ACC plus, absence of the corpus callosum plus other developmental brain abnormalities where typically lower functioning have more developmental delays. So that's the group that I will be talking about in this talk. Dr. Paul and I have done a lot of years of work, and a couple of years ago, we decided we would try to formulate this into a model that provides a structure for thinking about what are the consequences of agenesis of the corpus callosum in this higher functioning group. And of course, the diagnosis is agenesis of the corpus callosum. That's what we start with. And then we looked at the research we had done up to this point and asked ourselves, is there some summary core syndrome that we might extract from looking at the outcomes in these studies? And one area is, in some sense, definitive and somewhat obvious, and that is limited interhemispheric transfer of complex sensory motor information. Some information crosses, but complex information is not likely to cross, and we have studies that demonstrate that. All of this, by the way, I will elaborate more completely in my talk tomorrow. And then the second one is slow cognitive processing time. That is, you just process information a little more slowly. This is a syndrome also found in things like Alzheimer's disease, where people begin to just process information more slowly. And a third one is deficient processing of complex and novel tasks. So when you have a problem-solving task, and this is new to you, and it's a complicated task, persons with HCC just have a harder time getting there. They may get there or they may not, but it's much more difficult. So those are the three areas that we think are something like the core syndrome. And those kind of come together and combine in various ways to present what we call secondary symptoms of agenesis of the corpus callosum. So let me give you just some examples, some thumbnail examples of what we might consider secondary symptoms. Things like inadequate encoding of complex new memories. When you encoding is getting the information in there to start with. And to get it in there to start with, you have to encode it, to elaborate it, think about it in ways that connect it to other things such that you can remember it later. Failure to utilize all previous information in a problem-solving task, particularly serial problem-solving tasks where you have to move information forward. When you get to step 3, you've got to remember what happened in step 2 and step 1, and often persons with ACC just don't bring that information forward. So when they're at step 3, they're just at step 3, and step 1 or 2 might not have existed. Difficulty understanding the secondary meaning of language. Language always has a concrete immediate meaning, and then many times has secondary meanings. So idioms are statements that have secondary meanings. Metaphors obviously have secondary meanings. Jokes, proverbs, stories, sarcasm, all of these things have more than the immediate linguistic meaning. And individuals with agenesis of the corpus callosum often and sometimes don't pick up the secondary meaning. And so they're kind of stuck on the concrete meaning of the words and statements. Deficits in understanding the subtle aspects of social interactions. Social interactions, again, like language, have immediate implications, but really to understand what is going on, you have to imagine what might have happened before, or take what might happen before and use it to understand this situation, and to make some sort of predictive imagination about what's going to happen next, so that you can track the social interactions in complex and nuanced ways. And then difficulty understanding the consequences of possible actions. So I'm in a situation, there are several possible things I could do. I need to be able to imagine the consequences of this action versus that action in order to make an adequate choice. And that's sometimes difficult to figure out exactly what what are the consequences of doing X versus Y, for example. So those are the secondary symptoms. And what I want to do primarily in this talk is focus a little more on the deficits in processing of complex and novel tasks, and use some recent research that we have done to kind of elaborate and expand on what that might mean. So I think there's this is just my thinking about this, some related context. So we have the idea of problem solving, complex, novel problem solving, a difficulty we know from a number of different experiments we've done are difficult for persons with ACC. But to do problem solving in a complex task, you have to imagine what might be the case or could be the case if I begin to work with this problem in particular ways. You have to imagine possible steps in the problem solving process. You have to imagine what other information that I know might be related to this. So it takes some imagination, and imagination takes elaborative thought, inference. You have to take what's available now, elaborate on it, and infer what might be the case beyond what's immediately presented to you. But this idea of imagination also, I think, relates to creativity, not just problem solving, right? Not that those two are easily distinguished. Problem solving in complex novel tasks takes a certain amount of creative imagination in order to do that. So what I want to do is present some data that I think reflect elaborative thought in one particular context to suggest how this might work out. So I want to ask the question, do persons with ACC have difficulty thinking beyond the information that is immediately available? That is, do they have difficulty thinking imaginatively and elaboratively? So we have this test we did. It's a test of imagination. You look at little videos of little triangles moving around, and you merely say, after the end of the little animation, you just say what I saw. This one's kind of random. There are also ones that are like more goal-directed. Simple inference. One is trying to follow the other. So but the ones we are really interested in are ones that cause us to think and infer some things that might be the case given the interactions of the triangles. So let me show you this one. And for you to understand where we're going with this, I'll show you the whole video. It's only a couple minutes here. So having viewed that, the task is, what did you see? And you and I and most people would describe something that is full of content about social interactions, mental states, emotional states, social interactivity, and we would describe it that way. We got this test from persons who were studying autism, and the result with persons with autism is that they really leave out all of that social, emotional, cognitive information and pretty much tell you where the triangles went and how they interacted in that way. So we've been very interested in this test. And so we have data on it, and the response is a text. So if you take what they said and you transcribe it into a text, you have a verbal written statement that this person said. And so we were interested in applying new methods of computational linguistics with respect to the statements that the people made. Computational linguistics is a whole area developing in which they use computational methods to try to analyze text to pull out meaning out of text. And so we used a model called topic modeling. And topic modeling takes a text and tries to extract from it the primary topics that were in that text. So what we did is we had individuals, neurotypical control individuals that had a corpus callosum, and we took, there were 14 of them, and we took for each, there were 4 different animations like you saw. For each animation, we took all the controls and we lumped them into one big text. And that was kind of our core model text of all of what persons with a corpus callosum would say. And then there's a way in topic modeling to take a model from a different model and compare it to the first model. So what we did is we took the model of the statements made by each of the persons with agenesis of the corpus callosum and compared them to this core model. And there is a statistic that is calculated that says how different are the topics in these 2 texts. And if it's very different, it's said to have high perplexity. So we had a perplexity measure. So topic model of ACC person 1 was compared to the overall model, and then for person 2 and person 3, and we got a perplexity value for each individual. Then in this test, we also had individuals with autism spectrum disorder. So we took each of the persons with autism and compared their responses to this core text. And then, of course, we had the control group itself that comprised the core model. In this case, we took out the person from the core model that we were comparing, so you weren't comparing them to themselves. So it took a whole lot of calculation intensity to do this part of the work. And so we have perplexity values for persons with ACC, for persons with autism, and for persons with normal persons with a corpus callosum. So would you like to predict how this comes out? Where the perplexity values, where the persons with ACC have very similar or very different? Low perplexity, very similar. High perplexity, very different. Anybody want to vote for high? Anybody want to vote for low? I asked that because I actually got this wrong. When I was thinking about this before actually seeing the data, I kind of had in my mind that it would go different than it did. So here's the outcomes. There are 4 different animations. The one we saw was the surprise animation. So we can look at this. When the blue bar is the individuals with a corpus callosum, the controls, and they have very high perplexity values, whereas individuals with agenesis of the corpus callosum and the individuals with autism had low perplexity values. That means that the persons with ACC gave a response that was just very much like the core response. But persons with a corpus callosum gave you the core response and a lot of other stuff. They elaborated and thought about and imagined a lot of stuff that would not be recognized as part of just the core, the thing that everybody says. Okay, so that was interesting. So this idea of elaborative imagination I became very focused on that as one of the important issues in these persons, high functioning persons with ACC that we've been studying. So after having done that, we had another data set, and this was from a classical psychological assessment called the thematic apperception test. And in this test, you just show one of these pictures, and there's a bunch of different pictures. These are just two of the pictures in the test. And you give a picture, these pictures to the person, lay it before them, and then you say, I want you to tell me a story with a beginning, middle, and end. Tell me what the characters are thinking, feeling, and doing. So you give them the thing and you write down everything they say and you transcribe it. And then you have it available once transcribed for computational linguistic analysis and topic modeling. So we did the same thing. This time we did not have a group of persons with autism spectrum disorder, but we had a larger group of persons with ACC and a larger group of controls. So we decided to see if we could replicate this in a different test, but with a similar thing. And sure enough, it came out very much the same. The blue bars are the persons with a corpus callosum, neurotypical individuals. The orange bars are persons with agenesis of the corpus callosum, and pretty much the same result from every one of the 6 cards. So, and then if you want statistics, the statistics on this and effect sizes are very robust. So in a previous study of that thematic apperception test, we actually had done a different version of computational linguistics called linguistic inquiry and word count, which is a process of counting words in semantic categories. And so the categories of words used less by individuals with ACC are words dealing with emotions and feelings of the characters. They just don't tend to tell you much about the emotions and feelings of the character. Words related to the mental processing of the characters, things that would be going on in the person's mind. And words related to social interactions. So the things that are not elaborated or not imagined, or that are imagined by the persons with a corpus callosum and not readily imagined by persons without a corpus callosum are things that having to do with the other person's emotions, the other person's mental processing, what they might be thinking about, and what might be the implications of all of this for social interactivity. So I think there's information here that elaborative thought and inference is a difficult thing for individuals with agenesis of the corpus callosum. Suggesting less imagination is one way you might think about this, showing itself in problem solving and creativity. So where we're going with this is, whoops. The implications for this, for those with broader cognitive limitations, this is probably a minor issue at best. That whether or not you're creative, if you have a more serious cognitive deficits, this is not important to think about. But for those with the ACC itself, without other complicating that are higher functioning, the implications or this might be things like ability to anticipate the consequence of actions and situations. If you can't mentally elaborate and imagine, it's much more difficult to anticipate situations. Capacity for recognizing subtle social dynamics and ability to solve problems in a domain that is novel for you, which is some of the things we had talked about before and we've seen in a number of other studies. So where we are going with this, so our next new directions is to look at empathy. Empathy is a way of understanding the situation of another person and what is implied by their current situation using the multi-factor empathy test. We're also interested in aesthetic appreciations in visual art and music, which are, kind of processing and imagining beyond the artwork, whether visual or musical. And we're also interested in difficulty in imagining interpersonal choices and using the moral dilemma scale. And so that's kind of where we are going from this. We are working in a network, as Dr. Paul said, involving my lab at the Travis Institute, Caltech, a whole bunch of colleagues in IRC5, and then always gratefully dependent on NODCC, OSDOC, and Corpol and other groups to help us in this research by volunteering and suggesting and whatever. So thank you. Questions? Yes.
See, these are the things that we get at meetings. That's very helpful. We've added a sleep questionnaire for the infants and are getting that, but we have not added sleep for adolescents. Easily done. We can gather some information on that. Yeah. Yeah, and we've got that in the kiddos. We're getting that, but or younger kids, but not going up. But that's a great question.
Yeah.
Sorry.
Yeah, yeah. There's not a lot of good work as far as I know on therapies. Huh? Oh, the question is, are there therapies that would be applicable or helpful in the context of what we're talking about with reduced elaborative thought? I really don't know. What I think I would do is try to help my person with ACC not as much as possible, not to be in a situation where they are, particularly for safety reasons and social reasons, needing to solve this problem on the spot. That is, as much as can be learned in a more rote way, so that this is not a novel situation. So for my money, I think I would do my best to reduce novelty as best I could with my family member with ACC, such that they know what they can anticipate what's going on and have already thought about what might be the best things to do when they get in that situation, rather than letting them go into novel situations, particularly that might have consequences for them without prior preparation. And for example, in school, one thing I've suggested in the past, get ahead of the curriculum with your person with ACC, so that at home, they're already have some acquaintance with what's going to happen in the class. So when it happens in the class, this is not new and novel and requires them to kind of imagine what might be going on here. They've already been able to get some acquaintance with that at home. But beyond that, we have a lot to think about, a lot to do.
The thing I would add to that, and that I like that example, Warren, that's great. The prepping ahead of time. You also want to remove the workload of the brain and the cognition. So anything that you can help become automatic means that they don't have to think about that. And there's more mental energy for thinking about what is new and what is different. And so starting very young, setting up patterns. I always use driving, we learn to drive and then we don't think about driving very much unless something happens. if you set up patterns and routines and we know I know where my keys are when I walk in the house, then I don't have to waste time thinking about where my keys are. So you want to do those types of things to simplify life so that the energy that is available can go to what is novel and more complicated.
Yeah.
But as Linda said, that's not scientifically proven. That's just, or it's even studied. That's just our inference from what we know otherwise, from the whys. Well, the mild moderate, I believe, was on Elliot's slides, and my understanding was you were basing that on, are they needing additional help in school, and how much help are they needing earlier in childhood and development? So someone who is not needing much help in school and are kind of keeping up would classify in that as, doing great, normal. The question is, when it gets to these more advanced things and the more complex things of adult life or transitioning into teen life, they may still not need help academically. They may still be able to do a lot of things, but daily functioning can become more and more problematic. So I often hear people say, oh, I know people that are perfectly normal and have never had a problem. And I'm not going to say they're not out there. And I always say, please introduce them. Definitely at Ostock, I've met some people that were well into their adulthood and functioned remarkably well. And here, all of them, though, would acknowledge pretty readily that there were things that they knew about themselves that they were always working to compensate for and deal with. And I'm always balancing, talking with parents. I don't want to be discouraging because it's amazing how much the brain can cope and do and how well somebody can perform under optimal conditions. But I think it's always important to recognize, and this brain is having to do it through unoptimal systems. And so the vulnerability to fatigue effects, the vulnerability to sleep deprivation, to hunger, to all of those things that would affect any of us. This is a system that's already working with little margin. So respecting that in your child or in your adult, in yourself, is something that we talk about a lot in the adults groups. And they'll say, yeah, I've had to learn how to handle that and not expect myself to function at my best all the time, because then I set up expectations that people get frustrated with. And so, how we describe what's optimal, I think, is important.
I'll just add one comment to that. Linda gave us a really good thoughts about the great variety in the population we're talking about. And for research purposes, you have to take this sort of fuzzy, long, big variety and draw a line and say, this is the group we're going to study. We draw our line at a IQ of 80. So what I've talked about are persons who would test at that level or above, but that doesn't mean it only applies to them. And then it's a moving target as to how much this applies to whom. So
I think we're out of time. Linda, do you want to add any closing comments since you kind of got us started on the
Yeah.
big picture?
Just to say thank you so much, all of you, for coming and hearing about the amazing research that is going on around the world. And thank you to all the speakers who really put in a great effort, those who are also on Zoom. I think just to close this out, this is a collaboration. It's a collaboration between families, individuals with disorders of the corpus callosum, doctors, scientists, therapists, all of us. It's a collaboration. And, you know, by working together, we can really make a difference to understanding the brain, and we can make a difference, hopefully, to the lives of people with disorders of the corpus callosum. So thank you all very much. And we look forward to interacting with you over the next few days.